guess we should start. It's my pleasure to welcome Neil. He's going to talk about theoretical model for persistent and oscillatory cell motility.
Okay, so Anthita, thank you very much, and thanks for the invitation to come here. And I will tell you about the work that the first part is already published recently, a few months ago, and that deals with the persistent part of the title, and the oscillatory part is something which we are still doing now and is unpublished, and I hope I'll get to it if I run quickly enough, persistently. So this was a collaboration that started actually 3 years ago when I was on a sabbatical in Institut Curie in Paris. Mathieu heads an experimental laboratory there about cell motility, and these are the people in his lab who did the work, and he's collaborating with Michael Sixt in Vienna. Raphael was the person with whom I did the theory, and Jean-François was Raphael's PhD student at the time, and he did the actual calculations. We just thought up the model. And the paper, as I said, recently was published in Cell, which was very nice to get into this very biological journal with a paper that has relatively lots of theory and mathematics in it. Okay, so the first indication that something interesting could be happening relating persistence and speed velocity was the first cell race that was organized by Mathieu several years ago, you see in 2011. And basically what Mathieu was asking people from all over the world to send him cells that are the fastest that they can have. And people did that, and they sent him, and then he placed them on these one-dimensional adhesive stripes, and the cells are confined to move along these stripes, and they cannot move off them. And that allows you very easily to monitor their motion for velocity and persistence, although you saw one case of cell division, but most of the time they don't divide, they are just motile. And this was the lady who won the first prize. And they had lots of data on lots of cells, and you can see that using this one-dimensional setup, you can actually stitch together images from different times and get these very nice chemographs where time is going down and this is position, and you can get the velocity from the slope, and you can also get the mean times between switching of direction, and that gives you the persistence time. So you both get velocity and persistence time. And here are all the different types of cells that they checked, from the cells that they got from all over the world. And there was lots of data. Okay. And there was not a lot of correlation that they found in this mountain of data, unfortunately. But one thing that they did find, although again it looks very bad, but they did publish it already in 2012, was a correlation, a very rough correlation between the mean persistence path. So the persistence time was measured in length, which is again not the best thing because you have here also the speed, and the speed is also affecting the path length. So it's best to actually compare persistence time and speed. But there was a positive correlation. Each point here is a different cell type. So again, it's a very difficult thing to learn something from, but there was a first indication that there could be a positive correlation between how fast the cell is moving and for how long in time it will persist in moving in that direction. Now again, it might seem to you sort of intuitive for you that if you're walking slowly, you can change direction more easily, and if you're running, you are not likely to change direction very abruptly, because if you do that, you will fall and hurt yourself. But don't forget that you're in a regime where inertia plays a very strong role. These cells, when they speed up or slow down, they're not going to hurt themselves when they change direction, right? It's not like us people running and falling over ourselves. This is a regime where inertia plays no role. So why should there be a correlation, a positive correlation between how fast the cell is moving and for how long it is committed to moving in that particular direction is not trivial. Okay? It has to do with how the cell is actually mobilizing its internal components in order to make motility possible in the first place. And that was the idea. Okay, so we told them from the modeling point, and that I'm jumping a little bit ahead, and I'll get to the modeling in a second, but I want to finish with most of the data now, that in fact there should be an exponential relation between the velocity, the mean velocity of the cell, and the persistence time. So the time it is persisting in the same direction rather than switching direction. And when they then plotted data from a single cell type, so even in a single cell type, you have quite a large variability in its velocity, because the cell can pick up speed and slow down. Why exactly, you don't know, but there is this variability happening naturally, and they could relate when plotting this on a semi-log plot exactly as the model predicts. At some point, there is a saturation of the persistence time. It cannot go on and on forever, because I guess there's other noise events that happen. For example, the cell divides, cell has other clocks inside of it that are not related just to motility. So eventually, the cell did switch direction at a point where it was moving faster, but the model would have predicted that it would continue to persist for longer. Now, this is particularly a case where cells are moving along these one-dimensional stripes that you saw before. And again, it's easy to define persistence time and mean velocity during that period between switching of directions. Then they looked at cells that are moving inside one-dimensional channels. So these are like holes in which the cells are moving. And again, these are dendritic cells, which are cells from the immune system, if it means something to anybody here. These are cells moving in 2D. Now in 2D, the issue of how do you define persistence is a bit more tricky, because these are not just changes in direction, which are easy to define in one dimension. So what they looked here is the time it takes for the cell to change its direction of motion by 90 degrees. That was the rule that they used here. You can use different rules that it won't affect very much the results, okay? Slightly.
When you talk about persistence, it's about the time. It's the mean time moving in
It's the mean time of moving in one direction before you switch by 90 degrees,
so
exactly.
I'm talking about distribution of times.
So this is the mean, right, right, yeah. And in fact, you see that this might actually be faster than a simple exponential, because on this semi-log plot it's not exactly straight, so that could be an exponential to some higher power, right? So it could be in fact that tau goes like the exponential of v, but v is at some power which is larger than 1. So we actually get from the model that this is about 1, but it can be also larger than 1, which might be actually indicated here. And these are other cell types. Again, this is in 2D. In fact, this works extremely well. Again, you might complain very correctly that we don't have many decades of persistence to prove that this is a really true exponential, which you would ideally like to have when you want to prove that something is really going like an exponential. You want to have several decades, but this is what you have from these cells. They don't move over a larger speed range than between, let's say, 1 micron per minute and 10 microns per minute. They can't speed up to 100 microns per minute, so this is what you have. Even in 3D collagen gel, again, then the exponential part seems even more limited, but again, it seems not bad. And this is even in vivo, which means that they looked at these fins of these fish, and these fins are thin enough so that they are transparent, and in fact you can squeeze them between two glass slides. And macrophages, which are these cells of the immune system, which are very motile, can be fluorescently labeled, and they can be actually looked at while they are moving inside the tissue. So that was also very nice. This is from a group in Italy. There were many authors on this paper. It was a big collaboration. Okay, so what do you need to know about the internal machinery of the cell that is actually enabling these cells to move, to be motile? So what I think you need to know, that's what I will concentrate on, is this dynamics of actin. So actin is a protein of the cytoskeleton. I'm sure most of you have heard about it. It is labeled fluorescently here. This is why you see it. And you see that it's highly dynamic. It is polymerizing in the leading edge of the cell and then depolymerizing in the back. And that's why you see this wind of actin, which is moving from the front to the back. It's continuously polymerizing in the front and getting depolymerized at the back. You see that also the back is sort of contracting, and by contracting, it is pulling back its own tail. So whatever the cell was adhering to on the substrate, it is detaching and de-adhering by contractility in the tail, in the back. It's sort of pulling its own tail back, while the front is doing something else. It is protruding forward, spreading forward on this flat surface. This is a flat surface, so this is motion in 2 dimensions. Okay, so the actin flow is important, and you see that in fact there are 2 velocities here. There's the velocity of the cell, and there's the velocity of the treadmilling of the actin, of this flow of actin. And you see that they're not the same. In this case, the flow of the actin seems to be much faster than the actual motion of the cell. So which one of these flows is in fact the most important, and which one of these velocities actually correlates to the persistence time? And the model that I will show you again after I go through all the data relates actually the treadmilling velocity of this flow, this internal flow, which is really what is important for the internal machinery of the cell, rather than the actual resultant motion of the cell. And this was shown by Michael Sixt in his experiments. So he could change how sticky is the substrate to the cell. So you can see, for example, a chemograph which is along this line here as this cell is moving. So this is a cell which is adhering very well. So you see that the front of the cell is moving continuously, this dotted line continuously forward, very nicely. The cell is moving very smoothly and fast forward. And you see that each piece of actin which is produced at the front is essentially immobilized in the lab frame. So every piece of actin which is produced in the front is immediately stuck to the surface. And then another one, then another one. This way is very efficiently pushing the front part of the cell forward. On the other hand, you can have a cell which is on a very slippery substrate. So you see that the front is not moving forward almost at all, or very inefficiently. And any piece of actin which is forming in the front is necessarily moving backwards. So in the lab frame, it is moving backwards. But again, in the cell frame, the motion of the actin here and the motion of the actin here with respect to the cell frame could be very similar. And this is really the motion that we are interested in, because we're interested in how does this actin treadmilling flow affect the internal organization of the cell. Okay, this is the model. This is really what the model is based upon. And therefore, we want to distinguish between lab frame motion of the cell, where the cell is moving in the lab. This is really how we see that the cell is moving, to this internal flow of the actin, which we are actually interested in. Okay, I hope this Skype won't interfere too much. So after Michael was able to play with the stickiness of the substrate, he could actually have for each cell the cell speed, which you see in the lab frame, and the internal retrograde flow of the actin. And of course, the combination of both is the real flow of actin in the cell frame, in the frame of the cell itself.
By cell speed, do you mean the cell surface speed, or you mean the?
Eventually, over a long enough time, it's not instantaneous, but over a long enough time, it's the motion of the center of mass. So it's really the motion of the center of mass. Indeed, if you just look at a single point on the surface, it might have larger velocity fluctuations. So we don't look at that. We look at the real center of mass motion. This is what we call cell speed. And how do you follow the center of mass over the tracking? You could actually look at the whole contour and get the geometric center of the contour, or you can label the nuclei. Both things are being used. And you can see again that there is a very nice exponential relation between the persistence time and the actual cortical flow of this actin in the cell frame. Actually, it's a very nice exponential relation. He also looked at another way of defining persistence time, and that is actually the lifetime of the cell being polarized. So these cells can be completely unpolarized and therefore not motile. They can be very well polarized and therefore nicely motile in this direction. And they can be multiply polarized and then again not motile very efficiently. So what they looked at is the mean time that the cell maintains this polarized state before it is spontaneously losing that and switching to the unpolarized shapes, versus again the retrograde cortical flow, and again the exponential relation survives. You have a question?
The speed of the retrograde flow when you have unpolarized, what is?
No, you only define it when you are polarized. So you look.
Nothing at all?
No, you have flow of actin here,
Yeah.
but it is coming from all directions.
Yeah.
Yeah, but
Are you able to measure this thing?
yes, but this is not what is plotted here. What is plotted here is the relation between how long you maintain this state and the flow when you were in that state. So you're right, there is also flow in each of these individual lamellipodia, these structures, but what is plotted here is the actual flow when you were in the polarized state.
So
Yeah.
this way you could have a kind of baseline for later.
But just to tell you that you're right, and we now think that in fact this relation that we are proposing here for the whole cell could actually even happen on a local scale, so that if one looked at the lifetime of a single small such structure, maybe one could find again a similar relation between the lifetime of that local structure, which doesn't affect now the whole cell motility, and the local level of the flow, which was able to form at that region.
Mir,
Yes.
how polarization is defined here?
So the polarization is defined when the cell has a single lamellipodia. It's done by eye, and therefore it is also highly motile. But the polarization is defined here whenever you see the shape of the cell like that, when it has a single lamellipodia structure. So
With
it was done by eye
respect to
here.
the center of gravity.
Sorry?
With respect to the center of mass,
Yeah, I mean, that you have just a single such structure. It's done by eye, so I cannot give you a mathematical definition of that, because with the eye you see when that structure begins to deform and another one begins to form, and then you say, okay, up to here the movie is, that is the tracking of the movie that I was looking at, and that is the time when it's lost its polarization. And then you look at this distribution of polarization times, and it's roughly a Poissonian distribution, and you look at the mean of that, and that mean is what we plot here versus the cortical flow. Okay. So why do I tell you already for 15 minutes about the cortical flow? Because we think that this is the key to polarizing the cell, allowing it to be motile, that you saw from the movie. The fact that this flow is allowing the cell to be motile, that is not new. These people know that this lamellipodia is really what is driving the cell forward. But what do we think it's also doing? So if you think about this actin, which is polymerizing here and flowing back in the cell frame of motion, then whatever you have as a protein or any molecule that can have an on-off kinetics from these filaments that can attach even transiently and bind to these filaments, it will flow with the filaments away from the front and towards the back. So you will naturally get a distribution of the concentration for this protein, which is depleted in the front and has a high concentration in the back.
But what is C? The concentration of
Concentration, concentration of this protein that can hitchhike a ride on the actin. So this protein, which is affected by the actin flow because it's binding transiently to the actin, is depleted from the front and it gets concentrated at the back. How strongly? Depending on, of course, the on-off kinetics, the diffusion coefficient of this protein when it is free, and the actual actin flow. Okay, so that is then what you expect.
Regarding protein, they move like diffusion, Brownian motion?
Yes, so when they are diffusing, they just have a simple diffusion coefficient. It is a bit renormalized here to take into account the fact that you have this on-off kinetics. So it's in the limit when this on and off kinetics is very fast, then you can sort of renormalize the diffusion coefficient and the advection to take into account the ratio of the amount of time that you're on the actin and advected and off the actin and freely diffusing.
So it's together with actin transfer.
Yes, so this is the advection by the actin, exactly. And there is maybe some noise, of course, and there is no flux conditions. In the beginning, x equals 0 and x equal to L. So the cell for us from now on, I apologize, will be a one-dimensional object, although you saw for your own eyes that it's not, obviously. It's a three-dimensional object, but this is what we have at the moment. Other people are also thinking of more complicated descriptions, but yeah.
But also the advective motion and diffusion is the same order, or just a number or something?
So we want to be in the regime where the effect of this treadmilling on the concentration will only be such that this length scale that you can have from D and V, right, the ratio between them is such that it's less than the length of the cell. So if it is ratio between these parameters is such that you don't have any effect, then of course it's not interesting for that particular protein. It depends very much on the values of D, but D for most proteins is very similar. So what is D that is important is how much it is attached to the actin. And of course the flow of actin, because the flow of actin can go down to 0 and can pick up to very fast values. So this is really the interesting variable that will vary a lot. Yeah.
V is a scalar, is it?
So in this one-dimensional system, it is.
It's not a function of space and time.
No, that's a good point. Once this flow, this conveyor belt is assumed to be acting on these proteins equally well all along the length, which of course is not true because actin is polymerizing here and depolymerizing all along the way, eventually depolymerizing completely in the back. And therefore, the likelihood that you will have to attach to the actin is larger here because it's just more actin and less here. This complication we took out. Yeah, you're right. So there's a conservation on the total amount of this protein. We are looking at timescales where the production and gene expression is not affecting things. Yeah.
And the last term, the gradient of psi,
Yeah, so this is just a conserved noise. So you want to because this is a conservation equation. Yeah. And at steady state, there is a solution which looks like this. And you have an exponential where the length scale of this confinement to the back is exactly this ratio between D and V, as I said. And of course, there's a normalization condition where, again, V appears, and of course, the overall length L. Okay. If we look at experimental data, then you see that, for example, myosin light chain, which was made fluorescent, and that is a protein which attaches to actin, was seen to have a stronger and stronger gradient when the actin polymerization was increased. So it seems like this thing of sharpening towards the back, the more the actin is polymerizing, is indeed what people observe for proteins or parts of proteins that are known to attach to actin. So this is the experimental data that we have, again, from Michael's sixth lab, and roughly supporting this exponential. But don't forget that this exponential is a steady state solution. So I already sort of tricked you into telling you that you can in fact first solve this problem and only then solve the problem of the dynamics of V, because V will have its own dynamical equation. So in reality, we have two dynamical equations which we don't know how to solve analytically. One is for C and the other one is for V. C is affected by V, and V, I will tell you in a second, is affected by C. That will complete the feedback loop. To simplify things, we assume that this protein first gets a steady state distribution while V is changing. So the time scale for changes in V will be the slow process here. And to be honest, the division of time scales here is not so large. Maybe it's justified. I mean, that's what I will try to show you, that the data supports the results that we get when we do this decoupling. Okay, so what is the dynamics of V? So first of all, what is the steady state V? So assuming that we have some steady state concentration profile of C because of V, what would be the steady state V that is due to this concentration profile? So what do I mean by that? Imagine that this protein is a protein that affects by itself the flow of actin. For example, imagine that this protein is an inhibitor of actin polymerization. So when it is depleted in the front, actin can actually polymerize much more happily in the front. And when it is concentrated in the back, there is much less actin polymerization at the back. So now if you look at the overall flow of actin in your system, basically what you want to compare is polymerization from this side versus polymerization from this side. So the difference between the polymerization in both sides will give you the net flow in your system. I'll explain why the star in a second. Another option will be that this protein is myosin 2. Myosin 2 is contracting. So when myosin 2 is depleted from the front but concentrated in the back, it is contracting in the back very strongly, and that is pulling, that is giving an extra pulling force on the actin filament. So that's increasing the retrograde flow of actin. again, it's like a tug of war, because there is maybe some myosin in the front which is pulling in the wrong direction, and therefore you want to compare the difference between the two. You could think of more complicated functional forms that will link back this distribution of protein back to the treadmilling of actin, but the simplest one is just to assume that it's a tug of war, a sort of tug of war between the two ends of your one-dimensional hypothetical cell, the front and the back. There's essentially two sides to your cell, a front and back, and whatever is in the middle is sort of only affected by the ends. And then the distribution is just given by that. The stars is just to say that there is some chemical reaction involved with these proteins, either because it's contracting or because it's inhibiting polymerization. And any chemical reactions you can sort of describe by this Hill function, which just gives you a saturation. We took n to be 1, so no cooperativity of any sort, but you can play with high orders of n. Qualitatively, things don't change. So now you see immediately that you have V here, but V is also in all these C's. So you have to solve self-consistently this equation in order to get what is the steady state V, what is your V star. And indeed, you can see that as a function of this coupling parameter, for example, you can have a bifurcation, which means when this coupling parameter is very small for a given set of parameters that you chose, then the cell cannot self-sustain a polarized state. Let's say you even started with some non-uniform distribution of this protein. The V that this non-uniform distribution produces is not strong enough to maintain this distribution. So slowly everything decays to 0. Above a certain critical value of this coupling parameter, you can self-sustain a polarized state. So the V is changing the concentration. The difference in concentration gives you a finite V, and that is actually self-sustaining. And then it's increasing with V. Of course, it can be also positive or negative in this one-dimensional system. There's no preference to moving right or left. Okay. So the equation of motion eventually that we write for V is very simple. Ignore these parts, which are just noise terms. Essentially, we say that V linearly attempts to get to the steady state value that it wants to have, which is this V star. So it's essentially just a damped dynamics of V attempting to reach the steady state value with some time scale which goes like 1 over gamma. The noise term, well, there is one simple noise, which is just a noise term in V. So V has some noise in it with some amplitude K. This term comes from the fact that in 2 dimensions, we're actually looking at V, which is in the radial direction. So it's a polar coordinate. So we always have this problem with the cusp as V goes to 0. Now, there is another noise that we need to include, and that is the noise in C, which you asked about. So although C, we said we don't deal with dynamically, we always use just the C, which is given by the steady state value that responds to the V that we have. So V will change with time. With it, the concentration profile will perfectly match it always to have this nice exponential. So we don't deal with dynamic C. But the noise in C we still have to retain. So if we do that, we find that the noise in C has some other amplitude Kc, and is related also to the mean value that we have of C at each point in X. So it's this value. And now we have to take that back into the V star, because you remember that our V star was in fact dependent on the C's. So the C that we have in the front and the C that we have in the back. And if these are a bit fluctuating, there will be also a fluctuating value to V star, which depends on this noise in C. So all this long story eventually means that our nice equation for the dynamics of V that we started with, which was just damped dynamics towards the steady state value with some simple additive noise, is now complicated by another term, where you see there's another Gaussian noise term that comes from C, from the fluctuations in the concentration at the two ends. Because you remember, V is given by this difference of the concentration at the two ends. But if they are fluctuating a little bit, you have to add that as another noise term. But their fluctuations at the two ends is itself affected by V, because all these C's here have V inside them, because these are all these exponentials which have V inside them. So this is now a noise term, which, unlike this additive noise in V, which was just a simple noise term, this noise has V inside. So it's what people call multiplicative noise. It's noise which depends on the value of the variable itself. And that is shown here more explicitly, that the variance in V has a simple additive noise, and a part where we explicitly wrote this subscript of V to denote that V is hiding in all of these average C's, which are these exponentials.
So is it increasing function of V? Or?
It's an increasing function of V, exactly. So in fact, it is a noise that, and you can understand it because when V is 0, then the two ends are exactly identical, and then there's no difference between them. As V is increasing, the difference between them is increasing, and therefore also this noise term is increasing. And that actually can help you restabilize the zero velocity state, because if you are in a system where noise is increasing with your parameter, it's like the quietest place is here, and as soon as you move out of that, you are getting hit by a lot of noise, it pushes you back. It's a little bit like this example of the Kapitsa pendulum, where you can actually stabilize a pendulum which is upright by giving lots of vibrations and noise, which means that in fact it gets more out of this noise, more kicks out of this noise, as soon as it leaves the perfectly upright position. So again, you have a noise stabilized situation, and it's similar here. So at the end of the day, for 2 dimensions, we have the dynamics of V, which is given by some functional, which is this simple damped motion, and the noise, which is also V dependent, unfortunately. And theta, the direction in 2 dimensions, is just making simple diffusive motion. Okay. This effective force, which is appearing here, is actually all this long term, and it can actually, and this is the noise, and the noise has this multiplicative part. Okay. Now, from this simple equation, which is not bad, it's just a Langevin equation for the dynamics of V. So we are talking about the dynamics of velocity. We are always in velocity space. We're not in real space. This can be converted, as people always do, to a Fokker-Planck equation, which will potentially give us the distribution of velocities. We solve that, and we see that the solution is a simple exponential of this effective potential in V space. How does this effective potential in V space look like? Well, it has these nice shapes. Okay, so what do I mean nice shapes? When we are in a system where the coupling beta was too small, you remember that there was not enough coupling to sustain polarization. Cell is not motile, and this potential is in fact a simple harmonic potential. Forget about the cusp at 0, because that's again a peculiarity of these polar coordinates. Essentially in V space, the cell is not moving with a particular velocity in any direction. You just have a Gaussian of distribution of velocities around 0. When we are above this critical bifurcation, you suddenly have a particular V which is chosen. So the cell wants to have a very particular velocity. Again, doesn't care in which direction in 2 dimensions. So the potential looks like this sombrero head. There is another regime where again you restabilize the 0 point around 0. So in fact you can have a very well-defined motion with a well-defined velocity, but sometimes spontaneously, because of noise, jump again to a regime where you have lost completely your persistence. So this will be random walk, this will be persistent random walk, and this will be intermittent random walk, where in fact you have persistence and periods in which you've lost persistence, and you are again making simple random motion around 0 velocity.
It looks like noise-induced phase transition.
So here the noise, no, to go between these things, you have to change the real parameters. What the noise is doing is helping you to stabilize this 0 point, so it helps this phase have a larger region in phase space, but to move between these phases, you need to change the parameters.
Not intensity of noise.
Not intensity of noise.
But if I do?
If you do, then you will just make more jumps over this landscape. The noise will just, it's like heating up your system, but it will not change the landscape itself. Why is that related to polarization time? Because you can think of the cell being highly polarized here and having 0 polarization when it is here, because then it has no particular velocity going anywhere, right? So its treadmilling velocity is 0. This front-back difference went to 0. So how long did it take it to lose polarization and move from here to here? So this is like a mean first passage time problem. So you can think of cells always starting here and how long on average it will take them to reach the peak. And the solution to this problem, under some simplifying assumptions, is a simple is a logarithmic relation between this time and this depth of this potential at this position V0, which is this particular position at this V0 that the system has chosen. So in fact, it is this exponential relation between this time and this depth. So this is exactly what I wrote here. With this theta, for most parameters that we played with, is of the order 1, but it's not necessarily 1. It can be a little bit larger also. And maybe the data also supports it. So this is essentially what we get from the model, and that is why we went back in the beginning of the talk and asked them to replot their data in this exponential manner. And as you saw, I think it works quite well. So essentially, what do we have? We have that this double well potential, when you are polarized, gets deeper the faster you move. So if you're moving with this velocity, you have only this barrier to cross when you want to change direction. But when you're moving with this velocity much faster, you have a much larger barrier to cross in order to change direction. So a one-dimensional version of this is very easy to understand. So this exponential relation between the mean time it takes you to cross over the barrier and the depth of the barrier is simply related because the barrier depth is giving you also the position of this minima. As this minima is moving to higher velocities, you're also becoming a deeper potential. So that's really all there is to it from the mathematical point of view. From the physical point of view, you can think of a more sort of heuristic way of thinking about it is that the cell is moving faster. It has pushed more of these proteins to the back, and therefore it is more polarized, and therefore to change that and to go all the way back to something which is uniform and repolarize in the other direction is just more time-consuming, more difficult, but these are just words. Here it's actually given in exact mathematical formula.
But if you change direction, you don't have any potential.
Right, so this is a good point. So in 2 dimensions, you can actually move along this easy direction without actually losing polarization. So in fact, what we say is that this persistence should go exponentially with the velocity only for small velocities, only when this depth of this potential is in fact more like a graph that would have been drawn here rather than this deep graph that is actually drawn. So when the potential is weak, actually you will more quickly go up this hill and lose polarization before you have wandered around by 90 degrees, which is the definition of losing polarization in 2 dimensions. The deeper this is, you are much less likely to actually lose polarization completely, and therefore you are more likely just to wander around here. And if this diffusion just takes you a finite time to wander by 90 degrees, at some point you will reach this saturation. So as long as the persistence time is dominated by losing polarization, so really going up the hill, then this exponential should be relevant. But at some point, the time scale of just simply wandering along the easy direction by 90 degrees will be shorter than the time of losing polarization, and therefore you'll be stuck at that time. That will be your barrier for persistence. And indeed, in 2 dimensions, you see that more than in 1 dimension. So the full phase diagram is shown here. We have actually 2 parameters. One is beta, this coupling between this difference in the front and back and the actual treadmilling flow. And CS is another parameter which you all probably forgot all about by now, but it's actually appearing here. It's this definition of these C stars that appear in the 2 ends, and it's actually this thing. It's actually the saturation parameter that you have here. So it's this saturation parameter. Okay, so that's a CS. So in fact, we have 2 parameters, this CS and beta, and you can see that if the coupling is very weak, then you're in this random walk potential, and you're just doing random walks, and you can, for example, plot here the effective diffusion coefficient of your cell. If you're above a critical beta and also above a CS, then you're in a highly persistent regime. and then you can plot, for example, here, what is your this V0 that you actually choose, this V star shown here, so you're moving to faster and faster velocities. And you can be in the regime where you are restabilize this 0 point, and therefore you're in this intermittent regime, and you can plot, for example, the mean time that you are persistent versus the amount of time, the fraction of time that you're persistent versus the fraction of time that you have lost persistence and got yourself restuck in this local minimum. When you actually simulate the trajectories of cells, if you simulate the trajectory of a cell under these conditions, it makes random walk, and look at the x's here, they are very small in these units. When you're in this regime, you make persistent random motion with a well-defined velocity. You're just wandering in direction space, and you see that the scaling is huge now because you're zooming past on this two-dimensional space. When you're in the intermittent, you see that you have stretches where you're moving very persistently, and then regions where if you blow them up, you again lost persistence and you're moving in a sort of very random motion around this minimum. How do these simulated trajectories compare to real trajectories? So you can find, even within the same cell colony, under the same conditions, cells that are moving like random motion, cells that are moving very persistently, and cells that seem to transition between persistent and regions where you see that you can blow up these parts, and you see that they have again wandered to become something which looks like random walk. Why do cells have this variability in nature, even under the same conditions, and they're all clones of the same single cell? This is, of course, the noisiness of this system. What is more telling, I think, is that if you look at the distribution of the velocities, this P of V that we can calculate from the model, then for the random walk, this is the simple P of V, which is just a Gaussian in polar coordinates, and you can compare the data to the theory. When you're moving persistently, you have this clear peak at this velocity, which was chosen by the cells, and again, the data supports such a distribution. And when you have this intermittent regime where you are transitioning between persistent motion and periods of time when you have lost persistence, you have this second peak. So you have a double peak distribution where you have a small peak around 0 and another clear peak when you're moving persistently with a very clear velocity. So I think these velocity distributions are in fact the nicest demonstration that you really have these three phases, and as the model says. This was appeared just similarly to just before us, I mean a year before us, and it shows also this strong correlation between what they call the diffusion coefficient of cells, which amounts to the velocity of the cells, and the persistence time of the cell. And again, there's a positive correlation between the two. This is the shape of the graph that our model would predict. And again, they didn't give here any model to why there should be a positive correlation between these things.
So just back to this slide. So cell migration doesn't follow random walk?
I guess, well, I didn't give this title, so I don't know what they meant, but I guess what they mean is that there is this correlation, that there is this interesting correlation between how long you're persisting in a particular direction to how fast you are. So there is some hidden correlation here, which is beyond what a simple random walk behavior would give you, or you would put in the model. So the conclusions, I want a few minutes to show you about oscillatory motion, which I promised you in the title, is essentially, I think, okay. So retrograde actin flow is playing a crucial role here, and I think this is something that should be added if somebody wants to model cell motility. And that a simple model gives you all the three observed classes, which I think is very cool because you have the random walk, the persistent random walk, and the intermittent random walk in one model. So I think this is not a trivial feat. Let me, yeah, okay. So this is the oscillatory part that I wanted to tell you about. So these are dendritic cells. It's a work of my master student Ido, which is, we're trying to write a paper now, and I hope it will be submitted soon. And this is an example of the dendritic walk, which is moving in this one-dimensional channel from Mathieu Piel's lab. And you see from the chemograph of this.
What is it flowing through?
It's moving in a channel, in a one-dimensional channel.
What's in the channel?
Nothing, just a medium, fluid.
Fluid, okay.
Yeah. So it's feeling a rigid. It's like a cylinder. It's like a hollow cylinder, and the cell is moving inside. So it's in front of it, it's pushing, you know, water essentially.
Sure.
And behind it, water is coming.
It's coming.
Yeah.
Yeah.
Um,
That's
Now you see from the chemograph that there are these oscillations, and this is the model that I will tell you in a second, if I have time. And what is also plotted here in the heat map is the concentration of myosin 2. So you see that in general, myosin 2 is really in the back, and that is not surprising because we said myosin 2 attaches to actin, and actin is flowing from the front to the back. So it should be one of these, what we call polarity markers, which gets pushed to the back, and therefore can serve this positive feedback that we propose. What you see, but which is interesting, is whenever the cell is actually decelerating, you see a large concentration of myosin which gets recruited to the front. While before there was less, the front gets reaccumulated with myosin. So this front-back imbalance gets a little bit lowered, indeed, exactly when the cells are slowing down. So
is there any way myosin is being produced? Why is it? How does it get transported to the front?
Okay, so this is what I'll tell you in a second if I have time. So let's close this and let's start this. Okay. What does it work? Okay, good. So yeah. So okay, so yeah. So this is the motion of the my computer is crashing. It's too much open at the same time. Let me close this thing. Yeah. Okay. Okay. So you cannot handle this? Okay. So you see them moving. And as I said, they make this oscillation. Let me jump to the model to explain to you what is happening. Okay, so what these dendritic cells are doing. So these dendritic cells, first of all, can be in the immature and in the mature state. When they're in the immature state, which is these movies that I showed you, what they're doing is that they're looking around for pathogens. They're looking for bacteria, for something that is invaded into your body. They are the sentinels of the immune system. So they are constantly trying to engulf the medium in front of them, and then afterwards analyzing what did they engulf. And by engulfing all kinds of structures, rubbish, pieces of other materials, they eventually can find something which is pathogenic. So they're constantly doing that. And in fact, you can see these vesicles that they are constantly creating at the front. You see? These vesicles. These vesicles get created in the front, and you can see that when these vesicles get created there is myosin which is recruited to these vesicles, and when myosin is recruited to these vesicles, the cells slow down. When myosin is not recruited to these vesicles, you see that these cells produce the vesicles, but they have been tampered with. There is this protein II, which has been knocked out. You see that they produce the vesicles, but they cannot recruit myosin there. There is a tiny bit of myosin there all the time, but it's completely constant. It doesn't reach the high values that you see here. The cell is actually unaffected. It moves persistently, just like I showed you before. Cells can move very persistently. So what is destroying this persistence is the fact that these vesicles, under normal conditions, this is the normal cell, get created, just like here, but also here they recruit the myosin to them. Why? Because these vesicles are in fact completely coated with actin. So actin is not shown here, but they are completely coated in actin. And when they are coated in actin, they recruit this pink myosin. This pink myosin helps to squeeze these vesicles. Only when squeezing the vesicles, the cell can actually analyze what's inside. So it needs this squeezing in order to actually understand what is inside those vesicles and then get if there was a pathogen there. But then this myosin which is recruited to the front comes at the expense of the myosin which was in the back and powering our forward motility. So we have a competition here for a resource which is needed for motility, which in this case is myosin 2, but it's also needed at the other end in order to squeeze those vesicles for the cell to actually do its functionality, which is actually to analyze what has it actually enveloped. And therefore, this competition gives you this periodic slowing down. This squeezing destroys these vesicles. There are no more vesicles. All this myosin can be released. It gets convected back to the back because there is some residual active flow. And the system actually picks up speed again. But now again, it is accumulating vesicles. These vesicles recruit the myosin, and so on and so forth. So this was their understanding from the biological point of view. And all Ido did was actually convert this understanding into mathematical formulation based on this model that I described to you before.
Do you think it's a convective dominated process rather than diffusion for the myosin?
So myosin is constantly convected to the back because of the actin flow and diffusing. There's always a component of myosin which is released and diffusing, and a component which gets stuck on the conveyor belt and gets convected. So, but the convection is always one way here.
Yes, yes.
And diffusion is, of course, in all directions.
Sure.
Yeah. So the recruitment to the front is through diffusion. Yeah. Okay. So the minimal model that we came up with is essentially to say there is some amount of vesicles in the front, which will introduce a new variable called SF, vesicles in the front, and its rate of creation will be related to the motion of the cell. So the cell, while moving, it's actually producing this thing in the leading edge. Then there will be some constant rate of decay of these vesicles, so they have a lifetime, and possibly there is even a faster rate of decay, which is related to the amount of myosin that they manage to recruit to them. So even if there is no myosin recruited to them, they have a finite lifetime, but they will not accumulate up to infinity. But this rate of diminishing is increased by another parameter times the concentration of myosin in the front. So you might say, you already now have two new variables. So you'll have 3 dynamical equations, SF, CF, and V, and the velocity, instead of the one that you had initially. But CF will be treated just like the CX, just like the concentration profile. It will be treated as something that reaches an immediate steady state value. So we'll have only 2 dynamical equations, one for SF and one for V. Okay, and how do we do this? How do we treat CF as completely being immediately at steady state? Just like we did before. So in fact, CF will have some on-off kinetics, and it is recruited to the front dependent on the amount of SF. And that means that it has a steady state solution which is given by that. So we will always assume that it is in this quasi-steady state. It is not a real dynamic variable that we need to keep track of. So there's only.
What is CL now?
So CF is the coverage by myosin of these front vesicles. So it's a parameter which is between 0 and 1. C is still this C that we had before. So the cell still has a one-dimensional shape between 0 and L, and it still has this exponential decay of C of X throughout the cell. But in addition to that, we have in the front a region which has a CF times SF amount of myosin which is stuck on these vesicles in the front. And it can only recruit them to these vesicles from the front. So there's only an on-off kinetics on these vesicles from whatever is available in the front from this distribution that we had inside the bulk.
Do you still have conservation?
Yes, exactly. That's the whole point. So there is now a new conservation equation, but it doesn't conserve only what was in the bulk, but also the amount which is stuck on the vesicles. And that is still the total given to us. Exactly. And the one in the bulk is still given by the same equation that we had before, only the normalization condition is different because of this conservation equation. Exactly. So at the end of the day, we have just one extra dynamical equation, the one we had before, which is exactly like you saw before, but there is another one for SF, for the amount of vesicles in the front. And that is given just by that. It's increasing with V. So there's really no nonlinearity here at all, apparently. The only nonlinearity is here. Only CF itself is a nonlinear function also. So it's a bit misleading the way it's written here. So this is truly a nonlinear equation. Okay, and with the addition of the conservation equations and the actual values for C. So in this phase of amount of vesicles and velocity of the cell, what do we have? We can have a solution which is completely persistent, so we can have a steady state which is completely static and stable. But we can also have an unstable steady state around which we develop a limit cycle behavior of oscillations. And you can see that we cannot cross into negative velocities the way we wrote it, so we cannot describe the way I wrote the model so far a change in direction. So we can only have a very strong slowing down, picking up velocity again, and slowing down again. I'll get maybe in the last minute to the case where cells actually do change direction periodically. So you can see in this nice animation that Ido prepared how you can move from the stable steady states, which are on this branch, to the unstable steady states, which give you this limit cycle. And that's just the Hopf bifurcation that many of you have seen in different systems. And he's plotting the actual location of the eigenvalues here, just to show you in the complex space. Okay. You can of course cross this transition by increasing, for example, this parameter kappa, which is the strength by which the vesicles are able to recruit myosin. So if they cannot recruit myosin, so kappa is very small, it's when this animation starts, you're completely moving persistently. And that is what was this I minus mutant, where it was moving persistently, it was making lots of vesicles, but they just couldn't recruit myosin, so the motion was not affected. As soon as you can recruit myosin, you move in fact to a lower amount of vesicles, but also your motion becomes oscillatory. Okay, so this is all of that. There's an issue here about timescales, and I think you can agree with me that the chemographs that he gets from the model and the experimental ones are very similar. What he has done here is actually plot the cell. So this part is the cell, which has a fixed length in our model, 0 to L. The amount of concentration of myosin C of X inside the cell. And the front part, what he has done, he has added here a piece which its length is proportional to the amount of vesicles, because that's really what you also have in the cell. These vesicles take space. So the more vesicles you have, it takes more space. We don't assume that there is any variation across this space. So for us, it's a single point in the front. But if you want to plot it realistically, you actually have to give this amount of vesicles some length. And then there's a constant concentration of myosin, whatever they recruited at that point. So you really see that when they get squeezed, it's also the time when they are also the most coated with the myosin. Okay, I'll jump over all of this. You can actually plot the amount of the velocity variation. This is the model, the myosin in the front versus the myosin in the back, the amount of vesicles, and you can compare it to the data in that particular chemograph that I showed you. The problem with the data is that even if you see such 3 oscillations, you can tell me, okay, Neil, these are 3 oscillations, but this is just by chance. This is not a truly oscillatory system. How can you be sure this is a deterministically oscillatory system? And to be honest, we need longer tracks to prove that. Okay, I just wanted to show you, what happens if you start close to this point. You're moving very persistently because you're slowly spiraling out, and then eventually you hit the limit cycle. So you will have a small oscillation, and then because there'll be nothing, and then there'll be a small oscillation, and then you'll hit the limit cycle. And what is nice, another way of reaching the limit cycle is actually starting from far away. So you make a large oscillation, and then you sort of overshoot, and then you go into this limit cycle. What is nice is that he found trajectories which actually mimic both. This seems like you're very stationary, and then there's a small oscillation here, and then you go into this limit cycle, and here you sort of make an overshoot, and then you go into this limit cycle. Again, you can view this as anecdotal, but he was looking at many such movies trying to get some data. Just to leave you with another puzzle is that some of these cells, in fact, actually change direction. Again, almost like a metronome, almost like an oscillator. These are mutants. They don't do that naturally, but it happens. So you have to say, okay, where's your model? Your model only showed us this periodic slowing down. So what you have to now allow is for the actual treadmilling, which you asked me if it has directionality. We assume that it always has the same directionality. you have to now assume that it can actually switch over. And that means that you have to assume that this distribution can actually switch to the opposite distribution, and that you can actually now produce vesicles in the other end. So you have to add another variable for the amount of vesicles that you're now producing in the other end, because that's another part, a real part of your object. So you have to describe what is happening there. So whatever you do, you have to complicate the system, because you have to now have the vesicles in the front and the vesicles in the back. And the vesicles in the front are produced only when you're moving in the front direction, and those are produced only when you're moving in the negative direction. So there's something here which looks like a step function. But essentially everything is the same, but then you now move in a three-dimensional space where you have velocity, which can now switch between positive and negative, amount of vesicles in the front and amount of vesicles in the back. So now your phase space is three-dimensional, and you can have a limit cycle which is looking like this strange H shape, and you can actually switch between directions. And without doing anything to the model, we found, look, it has a very strange behavior. You're moving very fast in one direction, then when you switch direction, you're first moving slowly, and then you pick up velocity. We looked at the data, and they see the same thing. You switch direction, you're first moving slowly in the opposite direction, and then you pick up direction again. Again, they don't have many such events and a lot of such long tracks as you would want to compare it quantitatively, but I think it looks you know, positive enough to be hopeful. And then, of course, you can plot all kinds of flow diagrams in this phase space, showing this limit cycle. You can have actually also a system when you have a limit cycle of switching directions. At the same time, you have a limit cycle which is just in one direction or the other direction with this periodic slowing down. So both can actually coexist in the same parameter range. So I think the competition, which in this case is between this production of vesicles, which has this long name of macropinocytosis, and motility for the same resource, which is myosin 2, gives the oscillations in the dendritic cells case. But I think such competition can happen in many other cell types. And I had a discussion yesterday with Rob Kay, and he told me from Cambridge, he told me that in dictyostelium, they think that actually there is a competition between exactly macropinocytosis and motility because of competition for actin. Because as I said, these vesicles also get coated in actin. So actin is also a resource that you need for moving, but now it's needed for another thing. So it could be that in general, whenever you have a cellular functionality that competes with motility for something that has to do with the cytoskeleton, actin, myosin, whatever you want, it's needed both for motility, but now it's needed also for something else. You'll have periodic slowing down because the resource is just not there. And then maybe you can have even deterministic oscillations. Of course, noise will also affect things here, and all I showed you in the model in the second part was without noise. So noise will complicate things as well. This was a paper that appeared a year ago, I think, which is also interesting. Here they destroyed the microtubules in the cell, you know, a system that I haven't spoken at all about, and they changed the cells from moving extremely persistently to moving in an oscillatory manner, really, but really so oscillatory that they even move less than one cell length. It's really like the cell is completely stuck like that. It's really not moving at all. And again, maybe our model can explain what is in here. They proposed a completely different model, which is based on this LIGO, local inhibition and global enhancement idea. So, okay, and that's what he likes, Ido, to think about these dendritic cells that are like rust around. Okay, thank you.
We have 5 minutes of questions.
Yeah.
If I understand the model correctly, your C satisfies an elliptic equation, basically. If you're assuming it's always at steady state relative to V.
The C.
Yeah, the
Yes.
C.
Yes.
So you could write this down as an elliptic PDE for C, coupled to an ODE for V.
Yeah,
Basically.
Yeah, the PD for C we saw in the beginning. Yeah.
Yeah, but you don't have the time derivative. I mean, if I understand correctly, the simplification you're proposing says we just take an elliptic equation for C rather than we forget about. So we're always at steady state for the parabolic PDE.
Yes. In fact, when I entered Raphael's office 3 years ago, when I started my sabbatical in Curie, Raphael is not in Greece, he's in Paris 6. He wrote down he wrote the equation, we talked about it, we ended up with these two equations, and then he said, well, this is where we are stuck, we cannot solve, and the idea that I had was basically to say, okay, let's assume for the moment that we can decouple the time scales. Truly, the motivation behind this decoupling is not so strong. The time scale it takes for C, maybe this shows it all. Where is it? The model. Yeah. So the time scale for diffusion, here is not so faster than the time scale for the changes in the velocity of the treadmilling of the actin. So the time changes in the velocity of the actin treadmilling is of the order of minutes, and redistribution of this C is tens of seconds, or seconds to tens of seconds. So it's not a big separation of time scales. So to be honest, you have a more complicated dynamics here.
But you could solve numerically this
Yes.
system here without the steady state, and I guess that would be
Yes.
a comparison.
Yes,
with
that
the
would be a good
theoretical
comparison.
result.
We haven't done that, but that should be done. Solving this PDE and dynamically, and yeah, you're right. Please, if anybody wants to take the challenge, yeah. In 1D, it's a 1D system, yeah. It can be done, yeah. Sorry.
Right, you defined the persistent as the mean time of the cell move in one direction,
Yeah.
right? And so it depends on B. Have you got any statistics or distribution for this time?
So it looks, in one dimension, it looks like an exponential distribution. So like a Poissonian process, which is good for our interpretation that you're really in this double well, and you have this, Kramer-like escape theory for the time scale it takes you. So you have like a constant rate at which you are attempting, and you are successful on average only over a time tau. So
Yes, but then if I observe
because the noise is just the Gaussian noise, yeah.
Yeah. But if I observe the cell and your idea is that the faster the cell move,
Yes.
less likely change direction.
Yes.
So we should observe a very long distances for cells which moves fast.
Yes, this is a very good point.
And then this
Yes.
question relation to Levy-Flight, Levy-Ball distribution of distances.
Right, so the tail of the distribution, this is a very good point. We don't have it to a very high quality, like the quality you would want in order to distinguish between these fine points of Levy flights or etc. But and the problem is really just technical, because they have these stripes, and even to make long images, which they did try in order to get these good statistics, you have like 3 cameras next to each other in order to cover a long line, and even that might not be enough, because it might zoom one cell which is actually fast, as you say, and you want to actually get those fast cells in order to get this long end of the persistence times. They are the most tricky, and in fact you don't have, at some point you have a cutoff of your measurement. We try to ask them to work with longer stripes, because for the oscillations, for example, you cannot say that this is a deterministic oscillation with just 3 oscillations. You want to have 10 or 20 in a row, and then you can say it's a deterministic oscillator. So what they now think is that they can have a trick, and as I told you, you ask if they move in the tube and they have water in front of them, water behind them. You can have them moving in the tube, which is actually blocked. And then it seems that somehow there is a little bit of a pressure building up in front of them, and that makes them go into much faster oscillations. Why exactly? Maybe because they make more vesicles because of this pressure building up in front of them, etc. And that might enable them, on the same length of track that they have at the moment, with the same amount of cameras and not changing the whole lab, record maybe 10 such oscillations in a row. But again, now the problem is that as you're moving up this blocked channel, the system is actually not the same here as it was before, because the channel that you have still left is different. So
that's
other complications.
Sorry, the answer is negative. So there is no available distribution.
For the long
running
tail,
length.
no. For the long tail, no.
They can
For the
make-
part of the tail that you need in order to check this level flight question. Yeah.
They can make perhaps a circular track, going infinite time.
Yeah, that's not a bad idea. If you make it large enough, the radius of curvature, I guess, on the length scale of the cell shouldn't matter so much. Yeah, I guess with microfluidics entry points, which are too small for the cell to go through, you could actually all the time supply enough medium to make the cells happy.
Do you understand the role of the medium
In what sense?
in this process of the cell being able to
Inside the channel?
Yes.
Because outside of the channel, the medium, of course, plays no important role, right?
No, but
because it's
within,
an infinite.
within the channel.
Within the channel.
Does it affect the rate at which, because you're saying it's most very.
I don't think so,
goes
because
down.
I don't think they understand perfectly, because, for example, this observation that they saw that when it's trying to move up a block channel, at least that's what they tell me, I haven't seen it even in one movie, that these dendritic cells tend to start doing these oscillations on a faster rate. This was a surprise to them, so I'm sure they don't understand yet what is going on there. And is there really a pressure buildup, or what is building up that is actually telling the cell that actually it's moving down a channel which is not open, 100 microns down the line, but actually blocked.
Have they compared velocities in relation to channel diameter?
Yeah, so that's a very good point. So they did play with the channel diameter. Again, I've not gone through the data yet, but they chose okay, so what is the effect of the channel diameter? It does affect the distribution of actin in the cell, and it affects therefore the motility. So they chose to work in a diameter range which seemed to be most favorable for motility. If they make these dendritic cells try to move on a channel which constrains them much less, I think they move less well. And if they're constrained too much, at some point you're squeezing down the nucleus. And now these dendritic cells don't mind it too much. They have a very ultra soft nucleus, exactly in order to go in our body and look for pathogens, but still you're causing damage to the cell. So they found a regime where it works best. And exactly how does it affect the actin distribution between front and back, they don't understand yet. And I haven't seen the data, to be honest.
And so if I understood correctly, the behavior with the double potential, if you would define your polarization in the 2D as not being change in direction by 90 degrees, but say 45, you would have to observe a cutoff sooner in the
Yes.
exponential.
Yes.
Is that what you observed?
Yeah, I think they tried to analyze the data in this way, and they found it qualitatively. Yeah, I haven't seen a quantitative analysis of it, but yes, they did, which is really what you expect. If you would define it over a 10-degree change, it'll happen so soon that no way you'll have time to even have any dynamics in the actin polymerization rates or distribution of anything, and this will already occur. So, yeah, sure.
And the last question from me.
Yes.
If, let's say, the cell move and then with acceleration, then it means it's better, it's become more persistent.
Become more persistent.
So
Yes.
the question is, the velocity is constant, or it might be an increasing function of time?
So the velocity is constantly changing with time, right?
So it's increasing.
Yes, for sure, yeah. So the velocity is our dynamic variable. So the velocity is constantly, where is it? Yeah, the velocity is all the time. Your cell is in this potential in velocity space. So it might be with this velocity, but then it can increase to that velocity, right? Or decrease to that velocity. So it's constantly changing its velocity according to a time scale, which is this 1 over gamma, which appears there.
So again, if cells start to move,
Yes.
then the velocity first increases? Or?
Yes. You mean when you start off with the cells in the center here, so it has 0 velocity in x and 0 velocity in y, so it's completely stationary, and then it is moving down towards this velocity. It's accelerating, but we don't have any inertia here. We don't have I mean, you have V dot, but if V dot is such that you are approaching your steady state V with some time scale gamma. That is really what you have because of this linear damped motion. Think of V as like your X, as like your coordinate, like you usually think of it. In that sense, you don't have X double dot.
The mean time also increases.
Yes, and the mean time increases as you increase the mean velocity. Yeah.
I guess we'll join here and
I'm here until Sunday, so if people want to catch me for questions, email me and we'll set up a meeting. I think that's the best way to do it.